Okay, so this is the first audio lecture for this unit, and this unit is going to focus on foodborne illnesses. So we'll have some case studies, and we'll also look at some of the different outbreaks and some of the different common causes, infectious agents that lead to foodborne illness. So with foodborne illness, this is where a human is going to acquire an infection or injury induced by some kind of microbial agent, whether it's the actual microbe or whether it's a toxin it's producing due to consuming food or beverage. Okay, so there's a number of foodborne hazards, and so those can be chemical, physical, or biological. And so we want to keep in mind with these foodborne hazards that food is not sterile, so it can be beneficial, but we can also have pathogens or spoilers, and those can lead to foodborne illnesses. So foodborne illnesses can be a concern for the general population, but definitely for people who are immunocompromised, they are going to be at greater risk. So individuals who are very old or very young, so at those spectrums of age, for very young, the immune system has not developed enough to kind of fight potential infectious agents that lead to foodborne illness. And for very old, the immune response is starting to wane. So again, that puts them at risk. Also, people with chronic diseases such as cancer or diabetes, because of those, their immune system can be altered, either because they're under chronic conditions of inflammation or because their immune system's not able to function as normal. Individuals who have an organ transplant usually are immunocompromised because they're on immunosuppressive drugs so that they don't reject the organ. But because of that, it makes it tougher for their immune system to fight and recognize pathogens or foreign molecules. Then also pregnant women. So there is the immune privilege that happens with the fetus in the uterus, but there's also a lot of additional demands on a pregnant woman and from a biological sense, and so that puts them more at risk at developing foodborne illnesses. And then also individuals that are nutrient deprived, and so if they're nutrient deprived, again, that can affect how their immune system is able to respond. So with foodborne illnesses, there are infections that, again, arise from coming in contact with a food item that has pathogens on them. So again, traditionally with a foodborne illness, we would think about the living pathogen being there. So then the pathogen, that microorganism actually invades and damages the host because they have consumed it and caused some kind of damage. So just keep in mind that with a microorganism, if they're present in the host but they're not causing damage, we wouldn't consider that an infection. So in that case, the individual might be an asymptomatic carrier or a chronic carrier. But again, they're not showing symptoms. So when we're talking about an infection, this is where there's actual damage being done because the microorganism is trying to survive, right? So there's 2 points of view on this, the microbe's point of view, but then also the host's point of view. When we talk about foodborne illness, but really any kind of illness, we can talk about virulence. So virulence is the degree of pathogenicity that a microorganism has. Both bacterial and viruses can have virulence factors, and so this allows them to be more pathogenic, cause more damage, and increase severity of illness. There's also the infective dose, and that's the number of those organisms you would need in order to cause an infection. Just keep in mind that if you have someone who's immunocompromised, that infectious dose might be actually lower. So when we talk about an infectious dose, it's kind of the general consensus of like how many particles you'd have to be exposed to in order to get sick. But obviously there's a lot of other factors that are involved in that, especially from the host standpoint. And So you as the host, or whoever's the host, those factors that contribute to your overall health can also influence, your individual infectious dose. So it's not always the living organism that's going to cause the damage. We can actually have toxins that are being produced by the microbes, and that can lead to illness. So foodborne intoxication is where we have, again, this toxin being produced by the microbe, and that chemical that is consumed, that toxin that gets consumed in the food products. So again, it could be that the microorganism has been killed, but they've left behind a toxin, and so when you consume that contaminated food item, you would then become sick because of the toxin. Okay, so there are a number of different bacterial and also fungal microbes that do produce toxins that are of concern. So Clostridium's, Clostridium botulism, but there's other strains of Clostridium. Also, Staphylococcus aureus produces toxins, and Bacillus cereus. When we look at fungals, Aspergilla, Penicillium are also potential toxin producers. So then there's toxin-mediated infections. Toxin-mediated infections are referred to as toxo-co-infections. And so this is where you have a large number of live pathogens, but they produce the toxin and release that into the GI tract. So again, it's not the pathogen directly that's causing the damage. It's the toxins that are producing. But the production happened within the host, opposed to where we were talking about the toxin being produced. Pathogen is killed because of pasteurization or whatever other technique, and so then someone consumes the toxin. Now we're talking about, okay, pathogen is ingested, pathogen reproduces, and then produces a toxin and releases it, okay? And so, again, we have our Bacillus, our Clostridiums that can produce toxins, but we also have some of the E. coli's. So there's toxigenic and also hemorrhagic E. coli that produce toxins once inside, okay? Campylobacter and also Vibrio are also common toxin-mediated infections. So etiology is the branch of science that looks at the causes and origins of disease. Epidemiology studies various factors that influence the occurrence, distribution, prevention, and control of the disease. Epidemiology also is not just infectious disease, and same thing with etiology. So just keep in mind, a lot of times these two branches get grouped together into epidemiology. We'll kind of talk about epidemiology as a group and including the causes and origins, but also different factors that influence occurrence and distribution. So looking in the U.S., 38% of outbreaks have an unknown etiology, so we can't really pin it back to what was the cause, what was the source of the outbreak. So sometimes we can maybe indicate that going to the certain location caused it, or maybe eating this type of food caused it, but we can't always pin it back to a specific cause or source of that infection. So kind of what needs to be done to correct it. When we do look at outbreaks, there's about 31 different pathogens that are commonly identified in the U.S. Okay, PulseNet is a group of labs that use standardized testing methods. So they are able to DNA fingerprint the microbes that are taken from samples and then compare them to other cases. So PulseNet actually goes through and kind of does the contact tracing, if you will, the mapping to determine whether this is like a novel outbreak or whether these outbreaks are actually linked to one another. So if you had individuals who were in different states and had similar symptoms, and their samples were sent in, through PulseNet, you could potentially link those together, which then means you can maybe go to, okay, where did all their ground beef come from? And then, okay, they came from this, all the ground beef came from this processing facility. So PulseNet kind of allows for the standardization of the methods so that then we can be able to compare other cases. There are a number of different testing methods for pathogens. The traditional culturing that we've talked about to the more molecular and rapid methods that we also talked about at the beginning of the semester. So we do want to make sure that we have sensitivity, selectivity, accuracy, precision, rapid, usability, and cost. All those kind of factors play a role in what methodology is used to determine the pathogen that's causing a foodborne illness. There are more labs that are shifting from traditional culturing methods to rapid ones. This saves on turnaround time and also cost for like labor. There is some of the added cost of equipment and reagents, but again, that kind of turnaround and labor savings kind of balances out. And as we talked about at the beginning of the semester when we were talking about different methods, with culturing methods, you have to have someone who has more training because they have to be able to perform those culturing methods and those additional diagnostic tests. And then there's also some of that, when some of those traditional culturing methods and testing, there's a little bit of interpretation to it, where with some of these more molecular and rapid methods, it's a computer that's kind of determining is it yes or no. So that takes some of that human interpretation element out of it. We do want to keep in mind that no method for testing is perfect. There's limitations of every test, and so there's a potential for false positives and false negatives. And with the COVID testing, we're hearing all about this, but the reality is all tests are going to have a certain percentage of false positive and false negatives, and the idea is that those numbers are extremely low, and that in some cases, by repeating the test, and then taking the consensus of the results, that can lead to a more accurate diagnostic. And again, when we're trying to link like an outbreak together, especially when we're talking about different states or even different countries, having reliable data that's accurate is really important, and especially like in a timely manner, because obviously if there's an outbreak going on and you're having to wait weeks for results, that delays the identification of what's causing the outbreak. Right, so this is the intro slides or power audio lecture. There will be some additional ones looking at some of the different common bacterial, viral, and then miscellaneous category. So do look for those audio lectures.